Hello, everyone, and welcome to today's webinar, Human Proteomics from the Operating Room to the Lab and Back. I am Sabrina Lemus of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SomaLogic. To learn more, visit somalogic.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send, and we'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical questions here as well if you have trouble hearing or seeing the presentation. I'd like to now welcome our speaker, Dr. Vinit Mahajan, Vice Chair for Research, Director, Omics Lab, Molecular Surgery Program. Dr. Mahajan, you may now begin your presentation.
Thanks. Good morning, everybody. It's a great opportunity to speak to you about what we've been working on in the operating room and in the laboratory to help improve patient care, particularly for eye disease. But I think this will apply to lots of different human diseases that have some surgical aspect to them. I'd like to acknowledge members of my lab, collaborators, and our funding sources, which are largely through the National Institutes of Health. So I direct something called the Molecular Surgery Program, and in the last 5+ years in ophthalmology, we have had really remarkable changes and opportunities of how we diagnose and treat patients. From the illustration here, you can see we have the surgical ability to deliver any molecular therapeutic anywhere in the eye. I tell people we don't operate on tissues, we actually operate on cells, we operate on molecules. So in the top left corner, you can see us giving an injection, which is given in the clinic. It's given with a topical anesthesia. It just takes a few minutes. And these days, we're typically injecting antibodies. And these antibodies against vascular endothelial growth factor for patients with diabetes or macular degeneration control angiogenesis that's happening inside the eye. On the bottom left corner is a needle. This is a small 41 gauge needle. And I was fortunate enough to be one of the first gene therapy surgeons to treat children with a blinding eye disease. And because we can precisely deliver just a few hundred microliters of a gene therapy vector right next to the target cells in the retina, this turned out to be one of the first FDA-approved human gene therapies for anything. Now, one of the things that we haven't done or taken advantage of, which is what I'm going to focus on today, is not just delivering molecules, but actually removing fluid and looking at molecules. So, for example, if somebody comes into the emergency room with chest pain, we will draw blood, and then we'll measure a specific protein called troponin. If this protein is elevated, it's a sign that there's been damage to the heart tissue and that the patient's probably suffering from a heart attack. Now, we don't have these protein biomarkers for lots of human diseases, but if we had these protein biomarkers, this could help really inform diagnosis, operative therapy, selection of therapies, postoperative healing, design of clinical trials, and even drug design. And so as part of our molecular surgery program, here at the Stanford Byers Eye Institute, we're looking for that biomarker to help us treat and manage disease. Now, there's a major challenge in human proteomics, particularly with surgical disease, that I'll talk just a few minutes on how we've been able to address that. So this is an image of me in the operating room. We have instruments in the eye. This is a retina surgery. You can see the instruments going in, and then in the top, you can see a little white tube. And as we're working inside the eye, removing fluid, we're also infusing fluid to maintain the shape of the eye. Now, in virtually every single surgery, this fluid is removed and it's thrown away, which is a major missed opportunity for understanding patients' disease. So what we've done is set up a biorepository to help collect these tissues. Now, the operating room is really designed for getting a patient in and out safely and doing the surgery. It's typically not designed for high-level science, and that's the issue that we addressed. So one of the first things that we did was address the question of how do we get dry ice into the operating room? How do we do advanced molecular biology during the sample collection? And so we designed something called the Morley, or the Mobile Operating Room Lab Interface, which is essentially a lab bench that we put some wheels on. And the drawers, we have sample tubes, we have pipette men, we have a computer. Importantly, we have a dry ice box and tubes, everything that you would have in a laboratory. But now we can wheel it in and out of the operating rooms. So immediately, when we remove some fluid from the eye, we can immediately freeze it and capture information. This also includes getting the patient consent in the preoperative area, capturing patient data and information, and then putting the tubes into permanently etched barcoded tubes that are safe and appropriate for putting in a -80. Once this fluid is collected, it's then stored in a nearby -80 freezer, and we use a database to track each of those tubes in an XYZ system, so we can address the challenge of finding the patient tube with the disease that we need, which could be months later. More recently, we have now moved to a e-consent process where the process is entirely electronic, which aids in the efficiency of the system. So I'm going to start with one of our first examples. So uveitis is intraocular inflammation. It's inflammation inside the eye, just like you might have joint inflammation or any inflammation elsewhere in the body. And like any physician, we're challenged with, when we see this inflammation, what's the best drug to use? Now, I'll introduce you. This is a microscopic image of a patient who came in. We're examining them in the clinic, and if you follow this beam of light with the highlighted area, you can now see the arrow pointing to these white dots. These are clusters of white blood cells and leukocytes indicating inflammation in the back of the eye. Now, what was especially unusual about this patient is that he was part of a multi-generational family, a 7-generation family where half of each generation would have this kind of inflammation inside the eye, and half of every generation would eventually go blind. This was obviously a genetic condition. And one of the first things that we did at the University of Iowa was to stage the disease. Typically at around age 20, in the 20s, patients would come in with that inflammation that I showed. They would be developing a cataract, and then shown in blue, they would have edema of the optic nerve and retina. We would call this stage 2 disease, and it looked like any other kind of uveitis. And then within a decade, they would develop neovascularization, angiogenesis, and in this pigmented brown color, we're showing degeneration of the retina and photoreceptors. This would look like a genetically inherited photoreceptor disease known as retinitis pigmentosa. They would also start to develop some early scar tissue on the surface of the retina. And then by age 40, they would develop severe intraocular fibrosis, neovascularization, and then a retinal detachment, which is shown on the far right. This disease was named ADNIV, which is short for autosomal dominant, neovascular, inflammatory, vitreoretinopathy. We eventually identified the gene for this, which is calpain-5. It's a protease. But even knowing the gene doesn't give us a therapeutic, a clear therapeutic plan for these patients. What had happened over time is they had failed a variety of conventional therapies for inflammatory eye disease. This included oral prednisone, oral methotrexate, azathioprine, infliximab, and various kinds of surgery. So what we started doing was collecting fluid from the eyes as these patients went for surgery. Now, most of us are familiar with a conventional Elisa test. An Elisa test uses antibodies to measure a single protein. That's shown on the left. Now, once we get into proteomics, that means instead of measuring one protein, we're measuring 100 to thousands of proteins. And one of the first platforms that we used was an antibody array, shown here on the right. where basically antibodies to different kinds of cytokines are spotted on the array. And if the certain cytokine is present in the patient's eye fluid, it'll show up as a bright green dot. If it's absent, the array will look black. So we performed this for a number of our patients with this inflammatory disease. And this is the data generated as a heat map. Most of us are familiar with election heat maps where it's either red or blue, depending on Republican or Democrat, or purple, if it's somewhere in between. This is a nice way of showing quantitative data. And so in this case, if the square is bright orange, that means the cytokine is very highly elevated and expressed. If it's dark black or light green, that means it's low. Each row in this heat map represents a patient at various stages of disease, and each column is a different cytokine. And so this is remarkable in that now inside the fluid of the eye, we can now detect very specifically all the cytokines that are being expressed in specific the eyes of patients, and then at specific stages of disease. So one of the first things that popped out, which was just remarkable, was that there was no TNF alpha in any of the patient's eyes. So as I mentioned, one of the conventional therapies for patients with inflammatory eye disease is to use an antibody called infliximab, which is an anti-TNF antibody. It's given as an infusion to the patients on a biweekly or monthly basis. A number of patients had received this anti-TNF therapy, and they all had failed that therapy. It hadn't mitigated disease in any way. And now here we can see clearly in those inflamed eyes, those inflamed eyes are not expressing the drug target. And so why would you ever give a patient a drug when they don't express that drug target? In fact, that's sort of the conventional way of treating patients in a trial and error method. Now, one thing that we did see was we saw that there was lots of elevated VEGF expression. This is a cytokine that triggers angiogenesis, retinal neovascularization, and hemorrhages. And what we saw, and you can see on the far right panel, that at stage 2 and stage 3 disease, when the patients are having this angiogenesis and neovascularization, there's lots of VEGF being expressed. Now, at the time, a antibody against VEGF had started to be used for macular degeneration and diabetic patients that had angiogenesis. And so that seemed like an appropriate drug to try. Now, the normal way that we would treat patients if they came in with a bleed is that we would take them to surgery. The surgery itself would just trigger more inflammation, and then they would have a bleed later. So what we did instead of taking them to surgery is we decided to reposition Avastin, this antibody against VEGF. On the far left panel, where it looks all red, shows a patient with a bleed inside the eye. Their vision is count fingers. They can only see a few fingers. They can't read any letters on the chart. We gave an injection of Avastin, and then 2 weeks later, the blood started to clear. 4 weeks later, the patient was back to baseline with all the blood gone, and the vision had improved to 20/50. And so with the knowledge of proteomics of what cytokine was present, we were able to take what was normally a surgical disease and turn it into something where we could give an in-clinic injection of a specific therapeutic against a single cytokine and make a significant change in the disease. Now, one of the other things you can do with your proteomics data is you can take all that long list of proteins, feed it to software that begins to look for pathways, molecules that are linked to one another. And what we saw here is that there were a number of T signaling pathways being expressed in the patient's eyes. This correlated with several of the histological analyses. On the right, you can see lots of CD3 positive T cells in the vitreous of the patient eyes. We noticed two specific signaling pathways, the mTOR and the PI3 kinase signaling. And so we began to ask ourselves again, was there a therapeutic that we had on the shelf that we could reposition that might target these specific pathways? And what we identified was the methotrexate. Now, we've been using methotrexate injections to treat lymphomas in human eyes. Methotrexate was known to target the mTOR PI3 kinase pathway. And what we decided to do is when patients came in with this inflammation, that we would give them an injection of this methotrexate. And what the graphs show below, this is our initial cohort of patients, is that immediately after the injection, within a week, the number of leukocytes or inflammatory cells in the eye decreased dramatically. One of the things that didn't decrease or didn't change was something we call flare. This is the haziness or the fog in a headlight, which is representative of vascular leakage. That didn't change significantly, but now we had a drug that we could use to control inflammation. Normally we use steroid eye drops or different kinds of steroids, but in some patients that'll cause eye pressure issues. We also use this intravitreal methotrexate injection perioperatively, before and after and at the time of surgery, to help control the inflammation that occurs in these patient eyes with surgery. Now, one of the ways that the patients were being treated was with something called a Retisert implant. This is a surgery where we actually put this little device in. You can see the little white circle there that looks like a little aspirin tablet. That's actually a steroid implant that will have a slow release of steroid over 2.5 years. It's a way of locally controlling inflammation without patients having to take oral medications where a drug goes throughout the body and has potential systemic side effects. So now, interestingly, we give patients steroids and drugs all the time, but we never know what that actual drug is doing. So in this instance, in the heat map here, each column is a patient at different stages of disease. And we had patients where we had treated the patients with the steroid and were able to collect fluid before the steroid treatment, this implant, and after. And so this is one of the rare instances where you can actually see the proteomic signature of what your drug is doing. So in stage 2 disease, what we saw with the Retisert, the drug treatment, was that the T cells, the inflammatory cells, went away. In stage 3 patients, it actually controlled the angiogenesis. The blood vessels regressed and the development of new blood vessels was less. But in stage 4 disease, where there's severe intraocular fibrosis, the Retisert didn't have any impact on that. And on the right, you can see the heat map. You can see all of these cytokines that were elevated before the steroid. And then in panel C, you can see which of the steroids went down. We also collected data on steroids, rather cytokines that were unaffected by the treatment. So here, one of the interesting validations that we saw is that of the cytokines that were present at this stage 4 disease, there were no steroid sensitive pathways. And so frequently in these 2 images, you can see severe fibrosis developing. We call this inflammation and we treat it with conventional steroids. The patients always failed this therapy. And now we had a good molecular reason why they were failing the therapy, is that the steroid sensitive proteins just weren't there. Now, interestingly enough, what we did see is this cytokine interleukin-6 that was elevated in the patient eyes and remained elevated even after steroid treatment. This is shown in the top panel here, where you can see during the stages where there's more fibrosis, you see these bright green dots for the IL-6. You can see the TNF-alpha stays mostly the same. And IL-6 has become more and more popular these days and has been linked to driving fibrosis in patients in various kinds of diseases. So in the late stages of patients with this condition, once they had developed severe fibrosis and had this retinal detachment, which is shown in the cartoon on the left and is shown by ultrasound and optical coherence tomography on the right, where you have these retinal detachments, these are really surgically inoperable. You could spend hours removing all the scar tissue, reattach the retina, but the patient would rapidly develop scar tissue again. So we had a patient who was monocular. They had already lost vision in one eye, and we thought, well, why don't we try a surgery, but also add a proteomics-guided therapy. And this would include anti-VEGF to treat the neovascularization, methotrexate to help control the inflammation. But at this time, perioperatively and post-surgery, we would add an anti-IL-6 treatment, and we would reposition tocilizumab, an anti-IL-6 antibody given intravenously. The arrows in this image are pointing to some of the scar tissue where are the edges where we basically had to amputate severely fibrotic retina. But this is one of the first adeniv eyes not to develop recurrent fibrosis and recurrent retinal detachment after treatment. And we believe, although a single case, that it's really the molecular therapeutics that we added to the surgical treatment that made the difference for this eye. You can see some of the pigmentary degeneration. You can see some of the hemorrhages there. But this is the first time we're really controlling severe fibrosis in an eye. And so I call this the adeniv personalized proteome, where we really understand what's happening in the eyes of these patients. This is an unusual instance where an entire family, a group of patients has the identical genetic disease. It's almost like performing a mouse genetics experiment in the lab. It's about as close as we can get. And what we learned is that we can treat vitreous hemorrhages with Avastin. We don't need to take the patients to surgery. We can treat inflammatory cells with intravitreal methotrexate. We can delay disease progression with the Retisert. Steroid is not indicated for fibrosis. We can think about IL-6 therapy for fibrotic disease, and anti-TNF therapy is never really indicated. And so this is a big lesson to us about how much we could learn from liquid biopsies, fluid that we collected inside the eye, by doing proteomic analysis to understand the eye disease at a specific time, at specific stages, and then going after drugs that we have on the shelf and intelligently and rationally selecting those therapies. Now, I'm going to show some other examples in some additional diseases that are not genetic diseases and how we've been looking at using proteomics. So there's something called proliferative vitreoretinopathy. This is severe intraocular fibrosis, very difficult to treat, several clinical trials, but we really haven't figured out how to treat this. And so can we use proteomics to actually improve our clinical trials? This was published a few years ago. These are photos of some of the fellows and graduate students working on the project. This is a picture now of a 55-year-old, has a grade C proliferative vitreoretinopathy, and the arrows are pointing to just really severe fibrosis inside the eye. And the way that one surgically treats this is to actually go spend several hours with tiny forceps inside the eye, try to remove all this scar tissue, and then flatten that retina like wallpaper back up on the wall. Very challenging and not always a great outcome. So what we did is we played the same kind of game as we took patients at different stages of disease, collected their eye fluid in the operating room, and then performed different proteomic analyses on them, again using a antibody array. What we show on the left is a network diagram of proteins at early stages of disease, late stages of disease, and the proteins that really jumped out, in particular the mTOR pathway. And then what we're showing on the right is what I like in the middle here is the clinical phenotype, which is shown in black, where it's well established how doctors will diagnose and stage these patients, going from retinal tear to pigment deposition, retinal folds, and finally things called star folds. On the left, in blue-purple, are the specific cytokines expressed at each of those stages of disease. And then on the right are various therapies that exist that could be repurposed. Now, interestingly enough, there was a clinical trial using VEGF, an anti-VEGF again, and the trial failed. But what we found is that the VEGF is expressed only at the very earliest stages, but the trial was treating patients at the later stages of disease. So we think the reason the trial may not have worked is that they were using a therapy against a cytokine at the wrong stage of disease. So again, we think steroid-sensitive signaling pathways weren't represented, that steroid therapy trials have failed in the past. mTOR signaling and IL-6 targeting makes sense. You could choose sirolimus, you could choose those IL-6 inhibitors. And probably a better way to conduct clinical trials, especially when you have very specific molecular therapeutics, is to enroll patients not by just their clinical criteria, but whether or not they're expressing the proteins that you're targeting or specific biomarkers. Let's take a look at another real common event in our clinic, endophthalmitis, where we ask, is this patient's eye infected? So this is a 65-year-old gentleman, 6 days after cataract surgery. The arrows are pointing to this white fluffy stuff inside the eye. On the right, you can't see into the back of the eye. It's very inflamed. Now, you don't know if this is a severe postoperative sterile inflammation. We don't know if it's a bacterial inflammation. Because of the elderly patient and some other comorbidities, it could be any kind of infection, maybe a fungus, maybe a virus. The way we treat patients these days is we actually give them a full spectrum of antibiotic, antifungal, potentially antiviral. We remove a small amount of fluid, and then we actually send it to the path lab, and we put it on a Petri dish, and we wait for things to grow on the Petri dish to tell us what this might be. Well, what we did is collect fluid from a variety of eyes, sterile inflammatory eyes, eyes with infections, and you can see on the heat map on the left, there's clearly a protein signature that can tell us that this eye is infected or it's a sterile inflammation. And then in the Venn diagram on the right, you can see a bunch of proteins that very clearly tell you that this is likely to be a bacterial infection. It's a different kind of signature if it's a viral infection, and a different kind of signature if it's a fungal infection. So I think in this instance, being able to capture some fluid, performing proteomics can really tell us what we're treating very rapidly as compared to putting things on a Petri dish. So in the future, we're trying to validate these biomarkers prospectively, and we'd like to develop and design an ELISA for rapid diagnosis and potentially point of care. Now, in the ophthalmology clinics, we also encounter cancer. Our most common cancer is something called uveal melanoma. And like many cancerous diseases, we want to know, is this restricted to the eye, or is this likely to metastasize? So with these patients, we've also been collecting eye fluid and putting them under proteomic analysis. This is our team here. And in a pilot study, we collected tumors, these uveal melanoma tumors, from 8 patients. On the left-hand panel are photographs inside the eye, and you can see sort of brown, dark, elevated spots. These are the melanomas inside the eye. And what's typically done these days is a surgery that will biopsy the tumor, directly biopsy the tumor, which will damage the retina. It can cause some bleeding. Those tumor cells are collected, they're sent, and what's really a great piece of work done a while ago is that those tumor cells will be sequenced for specific markers to tell the patient, help tell us whether this tumor is likely to metastasize or not. Well, we wanted to test the hypothesis with that tumor inside the eye. It's probably affecting the proteome, the different proteins being expressed inside the back of the eye, inside the vitreous, and that's shown in this cartoon. Now, a lot of the proteins that are in the eye really stay sequestered in the eye, but they might leak out into the eye, into the blood, but they would be heavily diluted. So what we did is we collected samples of the vitreous fluid from these tumors when they were undergoing biopsy to see if we could identify biomarkers that gave us similar information. Now, what we're showing here in these heat maps are patients classified by the gene expression in the tumor. In panel A, this is called GEP class, and class 2 patients shown on the far right are patients that are more likely to have metastatic disease, where that melanoma escapes the eye. And what we found that's circled in red here is there's a very significant protein signature that correlates with that class 2 disease. So maybe it's possible that we can identify proteins and not have to directly biopsy the tumor itself, but just collect some of the fluid around it. Another signature that tells us, gene expression signature that can tell us if it's likely to metastasize, is something called the PRAME status. And tumors that are PRAME positive are more likely to metastasize. And you can see that there again is a pretty clear signature of a series of proteins that correlates with that gene expression pattern. And then finally, like we did in some of our first cases, we can identify very specific proteins where there are pre-existing drugs and consider rational anti-tumor therapy for patients, depending on whether they're expressing these specific targets. So for our proteomics signature for melanoma here, we really feel that we can probably distinguish these molecular classes with a simpler procedure. c-KIT and c-MET are potential targets for drug repositioning. And now we're looking at the blood and a different fluid chamber that we think may give us similar information. So let me switch gears now to some of the work that's been done by Julian Wolf, a postdoctoral fellow in the lab, and now looking at high-resolution proteomic profiling. So the images and studies that I've been showing you up until now are we've been biopsying the vitreous fluid compartment during surgery. As a retina surgeon, if the patient comes into the operating room, this is the kind of fluid that we're typically able to get. And when we remove that fluid from the eye, it's a chamber that includes about 4 cc's or 4 milliliters of fluid, and we can get about a half to 1 cc of fluid, which is sufficient for mass spectrometry and sufficient for some of the arrays, these antibody arrays that I showed you. But it turns out that there's this front of the eye fluid. It's called the aqueous humor. And in this cartoon, that chamber is highlighted in yellow. We can insert a small 31 gauge needle. The fluid in that chamber is about 250 microliters, and we can remove about 100 microliters safely. So it's a much smaller volume of fluid than we were getting from the back of the eye, but it's a lot more accessible. We can remove this fluid pretty easily in the clinic. You don't need to go to the operating room. But one of the real challenges here is the low volume that we're collecting. There is a low level of protein concentration. There are no cells there. But like I said, it's easier to access. So one of the most common questions that I'll get is that after showing data for back of the eye, the vitreous fluid, people will ask, can you find that same information in the front of the eye for front of the eye fluid capture? And so, one of the most common techniques, of course, for proteomics is using mass spectrometry. And the question is, could we use this in the aqueous humor? And so we had collected liquid biopsies from 3 patients while undergoing cataract surgery. And the aqueous humor, like the blood, has lots of albumin, it has lots of immunoglobulin and other proteins that can obscure the more interesting proteins when you're doing mass spectrometry. So we wanted to test different depletion methods. So we collected the fluid, performed protein depletion, and then used mass spectrometry. And what we found is that basically mass spectrometry could detect about 117 proteins in our hands. We tested albumin depletion, albumin plus IgG depletion, and another common depletion method, which removes 14 of the most abundant depleted proteins. And you can see in these Venn diagrams and the graphs, compared to undepleted mass spectrometry, that there was limited overlap. The depletion process actually removed a bunch of proteins that we could see in the undepleted proteins. And the overall counts of proteins that we would find were generally quite low. And in the bottom panel, in the heat map, you can see how the same sample, the identical sample of fluid, depending on your depletion method, had drastically different protein expression patterns. And so we really felt this wasn't a great method, although it's been used quite extensively, and I'll touch on that. So the next thing that we tried was an Aptimer-Based Assay, and this is from SomaLogic and their platform, to specifically look at Aqueous Humor proteomics, this small volume. And in this case, we had samples from 8 different patients while undergoing cataract surgery. This is we would consider this our normative control, meaning they don't have several of the diseases that I talked about before. They don't have inflammation, cancer, fibrosis, etc. Now, what's interesting about this Aptimer-based assay is that it has over 7,000 DNA-based aptamers, these probes, to detect over 7,000 different proteins. So it's array-based, but a pretty sizable representation of the proteome, certainly compared to some of the antibody arrays that we had been using. These aptamers are supposed to be highly specific and a pretty large library, and they have been optimized for quite an impressive detection range from the femtomolar to micromolar for specific proteins, purported to have low variation. And this gave us a platform to using a small volume of 55 microliters as the lowest volume, which is something we could certainly capture in our Aqueous Humor samples. And there's a cartoon of this aptamer array, or how aptamers are used to capture proteins. And so I'm going to just show some of the preliminary data that we have. And so on the left is a heat map, and this is quite remarkable to us. So these are 8 patients, males and females, ranging in age from 51 to 88, coming in for cataract surgery. And in the top half, you can see of that heat map, we could detect about 2,700 proteins in all 8 of the patients. This is an 8-fold increase in what we were able to see with our mass spectrometry methods, and our array methods are, have up to 1,000 proteins just on the array. But this was 2,700 proteins that we could detect in these patients. Below that, you can see another 2,000 proteins that vary between patients, and then finally 1,400 proteins on the array, but where none of the proteins were identified in any of these samples. Now, on the right, what we did is we did a literature search on all the Aqueous Humor proteomics previously published. In the gray circle, in comparative controls, there have been up to 321 unique proteins detected. In the right, in this blue-purple color, there's 1,700 Aqueous Humor proteins in the literature that have been detected in different kinds of diseases on all sorts of different platforms, whether it's LC-MS or DNA labeled or antibody assays. But in this SomaLogic DNA aptamer array, shown in green, we were able to detect close to 2,000 proteins in these normal eyes that had never been described or detected before. The physiology and the biology of what that aqueous humor fluid does is not entirely known. We know some of the physiological outcomes or purposes. For example, this is an immune privileged tissue, but we've never really known what are the proteins circulating in there. Now, the aptamer array did detect some of several, you can see in the Venn diagram, the overlapping areas, proteins that had previously been described either in control samples or disease. But right now, we think this is probably one of the best ways to explore protein expression in the aqueous humor. Now, one of the other questions that I typically get is, a lot of your diseases are happening in the back of the eye, and that's why you sampled the vitreous, which is shown in cartoon form on the left with that big yellow area. And that was all of the earlier disease data we were presenting. And they asked, well, can you detect those same proteins in the front of the eye? Because that's easier to get. And if you can detect proteins in the front of the eye, you don't have to go to the operating room every time. You could follow potentially the same patient serially, particularly with those cancer patients where you're trying to monitor for disease. So what we did is we took 12 patients where in the same eye, we collected the vitreous fluid in the back of the eye shown on the left, and we also collected the aqueous shown on the right, that small 55 microliter volume, and applied them to the DNA aptamer arrays. And these are patients, again, with non-inflammatory disease, not cancer, but were having surgery for one of the most benign conditions that we would say epiretinal membrane or macular hole, which we use as a comparative control. And what the Venn diagram shows is that of all the proteins expressed in the vitreous, which is in blue, 86% of those proteins are actually present in the aqueous humor. And so we imagine that 86% of those proteins are actually flowing back and forth between the two chambers. But almost as expected, 13% of the proteins are really unique to the vitreous. The vitreous fluid chamber is actually a gel. It has a collagen sugar matrix, and there are proteins that are presumed to be sequestered in that. So at least 13% of those proteins are only found in the vitreous. And if those are critical biomarkers, those 13% of proteins would require a vitreous biopsy, and you wouldn't be able to use them in the aqueous. But the vast majority of proteins are actually detectable in the aqueous, and I think that's remarkable and exciting. Now, one of the first things that we began looking at using this platform are age-related signatures, and so certainly in scientific circles, understanding aging at a molecular level and how one could intervene, since there are so many diseases where really the biggest risk factor is aging, people have been looking at specific protein biomarkers. A couple of those biomarkers are MMP10 and NRP1, and what we found is that those molecules are actually present in the aqueous humor, and in the panels and the graphs on the right, can be detected in the aqueous humor. And in our initial sampling here, actually show correlation with aging on par with the serum detection of these biomarkers. One of the really cool, interesting studies that has looking at aging, where they were looking at cerebrospinal fluid in young mice and identified something called serum response factor, which was depleted in the older mice, and replacing could turn the older mice and give them features of younger mice as a potential or anti-aging therapy. We could certainly detect that actually in the aqueous humor, and that actually had a very high correlation in terms of our human patients here as an aging biomarker. We could also identify several other biomarkers just specifically in the aqueous humor that correlate with aging. And now I think we can play the same game again of looking at biomarkers in the aqueous for various diseases that would help guide diagnosis, might help guide clinical trial and patient selection, could help guide determining if patients' cancer is likely to be metastatic or not, monitoring therapies, and actually selecting best therapies and patient responses to therapies at the molecular level. I think that's what we're looking ahead to now. And we can use the aqueous humor at an extraordinarily low volume and get really good data for making these decisions. So let me just show my last slide here. The challenge of the pandemic has been the separation of everybody. We're finally coming together in our clinics, in our operating rooms, in our labs. It was a time to reflect, and this is one of the slides I made. This is an extraordinarily complex pipeline. These are photos of the grad students, the postdocs, the surgeons, the technicians in lab, the bioinformatics, the proteomics team, the team taking care of patients and getting those consents, and then a number of the various faculty that we work with. Again, proteomics specialists, clinicians, and bioinformaticians. And I think if we can link our operating room to our laboratories, the opportunity to understand disease and treat disease is extraordinary, and should really be a focus of what we're doing. I think the different platforms of proteomic platforms that we have will really allow us to tackle a number of these challenging conditions. And while I've shown everything here about eye disease, I think this is broadly applicable to human diseases, particularly those patients that are ending up in the operating room. We don't want to miss the opportunity to collect that diseased fluid and perform scientific analyses that'll impact their care and treatment. With that, I think we have time now for questions, so I will pause there.
Thank you so much, Dr. Mahajan, for your informative presentation. We also have Claire Patterson with us now to help answer any of your questions. Welcome, Claire. We will now start the live Q&A portion of the webinar. So if you have a question you'd like to ask, please do so now. Just click on the Ask a Question box. It's located on the far left of your screen, and we'll answer as many questions as we have time for. So our first question I have here says, thanks for the great talk. And there's a couple questions in this one. Did your team ever consider an untargeted assay instead of targeted? And second of all, how were the control samples in the heat maps shown selected?
Sure. So we have used these three different platforms, and they all have their advantages and disadvantages. So one nice thing about this cytokine arrays is that they're quantitative. They're just like an ELISA, and so you can actually get picograms out of it. And one would, you can think that, well, path labs actually run ELISAs all the time, and so if you wanted to turn this into a clinically useful broad diagnostic, that pathway is pretty clear, but it's a very biased approach. And so the original arrays had a couple hundred proteins, and then now up to 1,000 proteins, but you're only measuring those specific 1,000 proteins. And so it is a biased approach. If you're looking at inflammatory disease and you want to know the cytokines, it's probably pretty good. But again, it's highly biased. The mass spectrometry approach is supposed to be unbiased. And so you're not just looking at cytokines, you're looking at, potentially the entire proteome, which depends on your mass spectrometry machine and how you're doing your liquid chromatography. But it's not quantitative, it's semi-quantitative, and there's different ways you're looking at that. People do the informatics for that. So you're looking at relative expression levels. And originally, if you find that comparison, you'll, on those two different platforms, you will get overlapping information, but it can be quite different for things just because of the methodology. And then finally, with the SomaLogic array, it combines some of the better aspects of both, in that you're up to 7,000 plus, which is a pretty darn good number for detection. You're getting close to the whole proteome. It's more unbiased than the ELISA. It's still not fully quantitative in terms of like an ELISA telling you picograms, and so you have that aspect, but it really allows you to tackle low volume. And so you can choose your platform, and they all have their own strengths and weaknesses. It depends a little bit on the question you're trying to ask, and frequently, we were using this a lot for discovery. Your validation experiments will sometimes use a different platform or different ways. And so it kind of depends the question you're asking, the disease you're looking at, and how you're looking at it for selecting the different platforms. But they all have their own strengths and weaknesses. And then finally, we don't take normal patients to surgery. And so we always have to use comparative controls. And our comparative controls for retinal disease are typically patients that have something called a macular hole or an epiretinal membrane. And in those instances, they do have some kind of disease because they're having surgery, but the clinical features are very, very different. And that's the comparison that most of us have done. And it's really important, that what we're doing with our databases is that we really have deep phenotyping of the patients. We have very specific stages of disease. We have their clinical data, clinical imaging, so that when we do put a group of patients together, it's a highly curated list of patients. I think that makes a big difference in the quality of the data that we get out of that.
Thank you so much for that answer. The next one that I have, given the change in proteomic capability, what do you anticipate in the coming years? For example, capacity, cost, availability?
Yeah, I think my general sense is, over the last 10 years, the accessibility to different kinds of proteomic technology has increased dramatically, and the costs have come down dramatically. The challenge with human proteomics is always this high quality sample collection. And so if you don't have high quality samples, or the patients aren't well phenotyped, or you didn't rapidly freeze the sample, you're junk in, junk coming out. A lot of clinical trials and clinical studies, for example, have deep clinical phenotyping, but not this deep molecular phenotyping. So I think that the challenge of just getting a good high quality patient sample is always going to remain the same. I think that hasn't changed. I think we've been able to innovate there, and the challenge there isn't really the science; it's really the administrative aspects of it. The quality of proteomics, and probably one of the biggest advances, is the accessibility of data analysis. So, the original data sets of analyzing the data and then what you could do with it was really complicated. But filling in that part of the pipeline, making the data analysis more accessible, having greater expertise. We certainly have a lot of expertise these days in gene expression analysis, RNA-seq, for example, and I think the proteomics will catch up with that. I think it's critically important. I always tell people, DNA is static. So we've done a lot of DNA sequencing. The chemistry is easy. Like I showed in my first example, the patients have a genetic risk factor, but that genetic risk factor or genetic component doesn't tell you the stage of disease, doesn't tell you the severity of disease. It really leaves out a lot. And certainly in the case of gene expression, again, where the chemistry is straightforward to measure, RNA and whether those genes have been expressed, the presence of mRNA still doesn't tell you whether a protein's being expressed, and doesn't always give you that dynamic effects of a disease in patients. And so I think proteomics is really incredibly important for the pipeline. The chemistry is really complicated. The informatics are complicated, but I think all of those steps have become more accessible. You can see from this last slide how many people are involved. I mean, it's a huge endeavor to run that whole pipeline. But like I said earlier, I think the opportunities are pretty amazing.
Maybe I can just add a couple of sentences to that. About the availability, accessibility, and cost, as well as proteomics becomes more accessible, which is fantastic. I think today's presentation is a fantastic example of discovery of our 7,000 plex platform at SomaScan. We do also have additional offerings of disease or biological process specific panels, as well as the opportunity to become kit sites where you can actually have the technology on site within your institution or company. So, SomaLogic is working towards that accessibility and increased availability of products to make proteomics more accessible, because we do believe, and today's a great example, that this is the way forward to help with biomarker discovery and drug targeting.
Awesome. Thank you for adding for that, Claire. This next one says, great talk. Thank you. Are there immune possible drug target signatures that cross between AH and VH that might have impact for sample collection and therapy delivery?
Yeah, so we're at the very earliest stages of really looking at this aqueous humor. As I showed in a slide, that at least in the, what we would call normalized, these are patients coming in for routine cataract surgery who were selected because they didn't have any other kind of retinal disease, for example. What we have to do now is look at the variety of diseases to answer those questions. But I think that's exactly the kind of question that we're asking. There are specific diseases where the inflammation is really restricted to the front of the eye. And this is called, anterior uveitis. And again, there's a diagnostic challenge where in some instances, this could be due to a virus, or is it autoimmune? Is it autoinflammatory? In those instances, I'm pretty confident that we're going to see pretty clear immune signature changes that could be measured to potentially guide treatment and monitor treatment. But I think what's great that we've seen is for back of the eye diseases in the vitreous, for retinal diseases, which is one of the, as our population gets older and older, these are some of the most impactful diseases on vision, impairing the independence and health of folks over 65. I really think that our opportunity to sample from the front of the eye is going to show us inflammatory signals that are originating in the back of the eye. So I think that's quite reasonable, and it's a matter of now of just collecting really good quality samples and seeing what's happened. The other thing that I didn't mention is we know that there are changes you can see in the retina that correlate with neurological diseases in the brain, and diseases that are systemic diseases will sometimes show up as molecules inside the eye. And so again, we're talking about eye fluid, but I think it's not just proteins coming necessarily from the vitreous. Some of those vitreous proteins are actually originating from outside the eye, and we may get some insight into those kinds of diseases.
Great, thank you. And I think we have time for just one more question. This one says, is the code you used for analyses of the SomaLogic data publicly available? And could you please point me to the manuscript that has that?
Yeah, we haven't. This is unpublished, really preliminary data. So hopefully we'll get it out there for things, and happy to discuss that as that comes out. And people are using this platform to look at other kinds of diseases outside the eye as well that are published, and I think would be pretty insightful in terms of methods and analyses and findings. I don't know if Claire had anything to add on that.
We have many publications in different disease areas, and we do have a publications link on our website that has a resource of all of those, many of which do contain methodology, and some of which do contain publicly available data sets. We also do have some software packages for reading and interpreting the SomaScan data, which is also publicly available, to make it easy for individuals who have assayed their samples to be able to interpret those results also.
Yeah, I think the analyses have the ability to do some basic analysis and see what's happening has become incredibly more accessible. That even if you're not an expert bioinformatician, that you can get some insight pretty quickly on those kinds of things.
Well, thank you guys so much for your time today and also your important research. We would also like to thank LabRoots and our sponsor, SomaLogic, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and their interesting questions. Questions we did not have time for today. There were a few that went unanswered. Those submitted during the on-demand period All those, they'll be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can also be viewed on demand. So LabRoots will alert you via email when it's available for replay. And we encourage you to share that email with your colleagues who may have missed today's event. Thank you so much. Until next time, goodbye, everybody.
Thanks. Take care.